Hello, I am Anuradha Malik. In the previous episode, we learnt about the cell cycle, wherein we studied about the various phases of the interphase. Today, we shall study more details of M phase. The M phase or mitosis has been subdivided into stages for convenience of study. The four stages are prophase, metaphase, anaphase and telophase. That cell division is a progressive process and very clear-cut lines cannot be drawn between these various stages. The first stage of mitosis following the interphase is prophase. Prophase can be marked by the following characteristic events. The chromosomal material which was not distinct and intertwined during the S and G2 phases becomes untangled and condenses to form compact chromosomes. The chromosomes are visible and they are composed of two chromatids attached together at the centromere. In animal cells, the centriole which had duplicated during the S phase move towards opposite poles of the cell. The nuclear envelope and nucleolus gradually disintegrate. Also, the Golgi complex and endoplasmic reticulum can no longer be viewed under the microscope by the end of prophase. Initiation of the assembly of the mitotic spindle occurs. The microtubules of the cell cytoplasm help in this process. Look at this diagrammatic representation of prophase. Notice the difference in the early prophase and the late prophase. The chromosomes have become shorter and thicker, and the nuclear envelope can be seen to be disintegrating. The next phase of mitosis is the metaphase. The complete disintegration of the nuclear envelope marks the start of metaphase. By this stage, condensation of chromosomes is completed and they can be clearly seen and studied under the microscope. The spindle apparatus is also completely formed. Metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to the spindle fiber from one pole and its sister chromatid connected by its kinetochore to the spindle fiber from the opposite pole. Remember that chromosomes at this stage are made up of two sister chromatids held together at the centromere. The plane of alignment of the chromosomes at metaphase is referred to as metaphase plate. Notice that in the transition phase, the centromeres have not yet reached the opposite poles completely. Also, the chromosomes are in a transient state and they are moving towards the equatorial plate. The next stage is anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split by splitting of the centromere. The two daughter chromatids, now referred to as chromosomes of the future daughter nuclei, separate, begin their migration towards the opposite poles. The chromosome movement is brought about by the shortening of the spindle fibers attached to the centromeres. When the chromosomes move away, the centromere of each chromosome is towards the pole with the arms trailing behind. Thus, diagrammatically, a typical anaphase is seen as shown in this picture. The next stage, telophase, begins when the two sets of daughter chromosomes have reached the opposite poles of the cell and the spindle disappears. The chromosomes decondense, uncoil and lose their identity as discrete elements. The chromatin material tends to collect in a mass at the two poles. The nuclear envelope assembles around the chromosome clusters. The nucleolus, Golgi complex and the endoplasmic reticulum also reform. Thus, in a way, telophase is a reverse of prophase. Each daughter nucleus now gets the same complement of chromosomes and nucleoli as of the mother cell. Diagrammatically, telophase would appear as seen in this picture. Following the nuclear division, which is known as karyokinesis, the cell is divided into two daughter cells by the process called cytokinesis, at the end of which cell division is complete. In animal cells, a cleavage furrow appears in the plasma membrane during telophase. This gradually deepens and ultimately joins in the center, dividing the cell cytoplasms into two. In plants, the formation of a new cell wall begins with formation of the precursor of cell plate between the two daughter nuclei. This grows from the middle towards the periphery and finally joins the cell wall. The cell plate represents the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. Sometimes karyokinesis is not followed by cytokinesis. This results in a multinucleate condition forming a syncytium. For example, the liquid endosperm in coconut. Finally, before we conclude this lesson, we must have a look at the significance of mitosis. Mitosis results usually in the production of two daughter cells with identical genetic complement. The growth of multicellular organisms is due to mitosis. It is significant for all repair, such as that of the epidermal lining and replacement of the blood cells. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. Cell division by mitosis restores the nucleocytoplasmic ratio. Look at another picture of mitosis as seen in a plant cell. This picture shows the images of mitosis as seen in actual cells on the right hand side and their diagrammatic representation on the left hand side. Students, you must practice drawing labeled diagrams of all the stages of mitosis. In the next episode, we shall learn about meiosis. Thank you.